Thank you very much, Wilson, for agreeing to give a Build a Cell seminar, and you can get started.
Oh, hi everybody. My name is Wilson Wong. I am a professor at Biomedical Engineering Department at Boston University. As you probably some of you might know, I do a lot of synthetic biology, a lot of mammalian cell synthetic biology. But today, I'll actually share one of our projects on cell-free, synthetic biology as well. We do a lot of circuit design and genetic engineering. Actually, I'm going to show some slides. I do have some overview on some of the stuff that we do in my lab. So okay, before I go on, just some disclosure, but for this talk, it should be okay. We do a lot of tool buildings for a lot of different applications now. It actually moves into a lot of different space recently. Traditionally, we do a lot of cancer immunotherapy, developing CAR T cells, cell therapy. But lately, I started moving into vaccine diagnostic and some tissue engineering as well. So today, I'm going to talk a little bit more about, let me see if I have laser pointer. Some of our work on, sorry, let me see. Okay, DNA recombinase. So we spent a lot of time designing how to create complicated genetic circuits using DNA recombinase, a little bit on tissue engineering, but also on cell-free controls as well. So I'm just going to move right in. I think, I like to think of my lab, we design a lot of widgets, for better or for worse. We do a lot of tool buildings and then apply them for various applications. So here it's a lot of receptor circuits, or these are CAR circuits. A lot of them are recombinases, and then lately we're using self-amplifying RNA as well. So we're making vaccines as well. So I like to think they're kind of like apps, and they have different applications and for in different settings. So I'm just going to go right in right now. Talking about one branch of our lab, it's on DNA recombinase. So we think DNA recombinase, it's a very powerful tool. So I'll tell you a little bit more what they are if you're not too familiar with it. So initially, we spent a lot of time doing these things with mammalian cells. We want a nice platform that will allow us to easily design circuits, and it can accommodate multiple input and outputs. And then it's robust, meaning that it doesn't require you to connect many different transcription units, different pieces, and then having them regulate each other. So we call them like minimize the number of layers, because that's usually the pain point in a lot of circuit design. So a few years ago, I'm just going to go through this pretty quickly, but it's been published almost like 8 years now. A system called Boolean logic and arithmetic through DNA excisions called BLAZE. You'll see this again and again. So just a quick reminder what site-specific recombinases are. So they are DNA modifying enzyme. So what you do is like you can use that to regulate gene expression, both to turn genes on or off. But then it doesn't work by a transcription factor, but it works by physically cutting pieces of DNA or changing the sequence of the DNA. So there are two ways you can turn things on or off. One way is if it's an on switch. What you do here, this is a promoter sequence, and then this is your gene of interest. What you do is that between the promoter and the gene of interest, you put a transcription termination sequence and what we call recombination sites right here. So you flank this transcription termination sequence. So in the presence of recombinase, what it will do is basically it will cut anything in between these two red triangle right there. So removing the transcription termination sequence, and then it allows you to turn on the gene of interest. And then similarly, if you want to turn genes off, you can, without changing the promoter, without changing the recombinase function, you can, instead of having a transcription termination sequence, you flank the gene of interest with the recombination site. And then in the presence of recombinase, it will actually excite your gene of interest, so it turns it off. So it has been really useful. It's been used in mammalian cells and in bacterial cells, and then animal model and all these things as well. So several years ago, we developed this platform we call Blaze. So basically, what it allows you to do is allows you to, in one transcription unit, to have fairly complicated circuit. So this is one of the most basic designs. So I'll walk through this quickly, but in a little bit more detail, because you will see this again and again throughout this talk in different settings. So again, you have your promoter right here, and then what you can do is you can place recombination sites. So we have the red one there for one type of recombinase called CRE, and then there's this blue one is for a different recombinase called FLIP. They are quite orthogonal to each other. And then you can arrange them in such a way that it creates 4 slots. Now, what will happen is that at least in mammalian cell, and also you can engineer in bacteria, in cell-free as well, is that the first slot is whatever gene of interest that you're interested in. It's the first one that it will get expressed. The other one behind it won't get expressed. You can have multiple genes, and then only the first one gets expressed, and they are separated by these recombination sites. Now, without any recombinase, this is the configuration of your transcription unit right here. Only that Z00 will get expressed. Now, what happened is that if let's say you have CRE, what will happen is that CRE will excise this piece right here, and then also excise this piece right here. Now, what it will do is that, it moves the second slot close to the promoter, and then now this second slot gets expressed. So sorry, the graphic is a little bit moved when I switch the slides a little bit. Now you have flip, and then basically the things between this green blue triangle gets moved. Now the third slot gets expressed, and then similarly when you have both CRE and flip, it will now the last slots, basically everything between the first and the third slots gets removed. Now the fourth slot gets expressed. So it's been pretty versatile. You can put a lot of different things in there. You can put, transcription termination unit, so it's a null space. You can put whatever output you want. We found that you can even put guide RNA as well. You can put 2 genes in there. You can even put more circuits in there as well, so you can make it even more complicated. So for more information about this system, you can see our paper in 2017. So one of the final outcome is this decoder is a 2-input, 4-output system right here. So this is the data point. Basically, you put 4 different fluorescent proteins here, you can get these output back there. So it's pretty nice, pretty cool. I like it. So I've been trying to always find ways to use it in different ways. Now, over the years, we've also been thinking about ways to control these recombinase activity as well. So right now, in all these experiments that I just shown you, it's either the cells either have the recombinase or doesn't have the recombinase. So it doesn't really sense any signals. But then these recombinases are actually quite useful. It's used in like mouse genetics, and so that you have these conditional and temporal control of gene expressions as well. And there were only a few inducible recombinase, and they mostly for one of the enzyme called CRE. So when we were designing our system a few years ago, and then we realized that there's not enough orthogonal inducible system to work with. So ultimately, we end up having to build our own set, and then eventually I might as well just build it out as much different system as we can. So we end up building about more like 20 different inducible systems with 6 different recombinases, and 3 different 4 different drugs, and then even with blue lights as well. So we actually tried it out in animal as well, and then showed that we can induce like gene expression like interferon gamma to cytokine productions. So these are old work, that's been done, a few years ago. So I'm going to share one of the work that we just published, like last month. Um, so it's about like, how can we use, how can we basically control uh, gene expression in a spatial and temporal way? Can you guys see? Is the I have these controls that block
We don't see that.
my
This is zoom controls that you see, but it doesn't come across on
okay.
the shared screen.
Oh, good, 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 good. Sometimes it blocks it too, a little bit. You see this white space. But anyway, so we're very interested in see if there are like spatial temporal control that ultimately, uh, let's say, change cell fate. Okay, and one of the things that we got really intrigued about, and there's a lot of people thinking about these things. Of course, is using light as a way to control gene expressions. There's it's a field called optogenetics, and the nice thing about optogenetics is that it has high spatial control. It's pretty fast in terms of like how it responds sometimes, and then also um you don't have expensive inducer. You just turn on some lights, and once you have the setup going, it's pretty easy. You can keep doing it over and over again. Um, so as you've seen, we had a blue light system, blue light inducible Cree earlier, and we asked, can we, just if just one light source, it's harder for us to have finer control and also like control different gene expression in different cells, or control like different genes in the same cell but at different time depending on the weight, depending on the light that we shine at it. So a lot there's only really just one uh, uh blue light systems available for recombinases. There are some red system that's available, and so first we started playing with it and see we can get them to work in our hands. And the thing that we like to do a lot when we do these inducible recombinase is that we like to split the recombinase into two pieces. Okay, there's an N-terminal piece and a C-terminal piece, and then we fuse them to these what we call light inducible dimerization domain. So what that means is that it will only come together when with light, and then at the same time, then it drags these two pieces of the same enzyme together, and then it will then reconstitute activity. We really like this approach. It's a little painful to use because you have to figure out where to cut the protein, but then it tends to, from our experience, have the biggest signal to noise ratio. It tends to have the lowest basal compared to almost any other inducible system that we have worked with, like Degron base or these ERT2, basically regulate translocation within to the nucleus. So this approach from our experience tends to be the best, but it's hard to use because you have to figure out where to cut. So and then, but luckily, when we were developing our inducible recombinase, mostly chemical inducible recombinase a few years ago, we screened through a lot of cut sites within, let's say, Cree in this case. And we have some pairs that we think it will work, and it seems to work for multiple inducible dimerization systems as well. So that's what we did here. We picked these sites that has been shown to work for other inducible dimerization systems from Cree, and then we start attaching to these three known red light inducible dimerization systems. So these are the paper that publish these things. So we want to see which one works with Cree. And then so that took a long time. Ultimately, I think the red map works the best. You can see here has the highest fold change. So in this case, in mag red, this at 105 split site, give you a little bit higher gene expression at the end, but then it has more basal activity. So what we learned is that even though you have these cut sites, you still have to figure out which system we haven't figured out any rules yet, which system works best with which cut sites, and there's always trade-off. Sometimes, there's some higher basal. But anyway, having multiple sites makes your screening is a little easier to work with. So ultimately, we decided to use RedMap. But then we also did it for Cree, and then we did the same thing for Flip. So we also found some, pretty nice cut sites for Flip. And then we try other enzymes. They are okay. They're not great. Here you can get like 50, 60-fold expression, but here's about a few fold changes. But that's the best we have right now. So it's nice that now we have blue line inducible Cree, red line inducible Cree, blue line inducible Flip, red line inducible Flip. So, okay, at least now we think we have, in theory, at least multiple multi-chromatic regulations of recombinases. So one of the things we're interested in is, okay, can we use our tools to influence cell fate? So we choose this model system. This is a fibroblast, the C10, C3H10 T1/2 is a clunky name. Basically, if you express a factor called MyoD, you can turn it into a fibroblast into a myotube. Okay, so it's pretty nice. You can see the morphology changes as well. So we learned that expressing GFP is a little easy, especially in hex cells transiently. But now if you try to influence cell fate, we need much better signal to noise ratio. And we found two problems with our red light system. Is that sometimes it's even though the dynamic range is pretty high, it's like 30-something fold, but sometimes it's just not strong enough for a reason that I'm not quite sure why. It just doesn't produce enough gene expression to, especially in this fibroblast cell line, to actually influence cell fate. So we end up having to build a sort of like an amplification system. So so this is a flex switch. So it's a little a little complicated. And what it does is that ultimately we want to drive, let's go on the right a little bit. We want to express MyoD. So MyoD, if it's expressed, then it will convert fibroblast into this myotube. Okay. Now, in order to express it, we want to it drives through a very powerful promoter. Okay, so that it expresses enough of this protein. So one of the most powerful promoter actually in our hand in mammalian cell is this P3 promoter driven by this transcription factor called TTA. So these are all synthetic promoters. Okay, they are more powerful than the CAG and the and the CMV promoter, at least in some cell types in our hands. So this is a very powerful system. Now, so what we want is basically having red light recombinase drive the expression of this transcription factor. Then it will drive this promoter to have a VP16 MyoD expressed. So the way we're going to do this is using this flex switch. So we did this also because we have to use lentivirus to introduce this whole systems into fibroblasts. So there's some complications with it too. So what it end up doing is that we have this two genes right here. One is M-cherry, so this is to make sure that the virus gets into the cell we can see. And then when we have the flip express, what it will do, it does two things. It does something called inversions and then excisions. So I'm not going to go into too much details into exactly how it works. So basically, it will rotate this TTA from an anti-sense direction into the sense direction relative to the promoter, and then at the same time excites this M-cherry protein. Okay, so it turns into something like this. Okay, so when we did this, we've shown that. So this is a pretty nice self-control system. We have a well with a photomask. One side is blocked, one side is not. So that when we shine red light into it, we can see like half the side it's illuminated with red light, half the side is blocked. It's a little bit of leakage into it, but you can see that and then you can count. We wrote a custom software basically count like when it's multinucleated, when it becomes this, elongated myoblast, and then we can tell that, this is, and the circuit it's on. So you can see a number of these few cells, changes by a lot when we have the light versus the dark side. So, so that tells us, we can now use light to, at least spatially control cell differentiations, even though we've seen some leakages as well. Okay. So next thing we do, okay, now we have red light, and then we know we have blue light as well. And then now we want to see if we can, having them use them together, okay, to control, gene expression. So, for example, let's say we want to build, this is just an example, okay? We don't really need this, but this is an example. Oh, there's some, can I answer these questions? I see some questions in the chat. Sorry, I haven't been checking.
You can answer right now, or you can answer at the end. That's totally up to you. Okay,
yeah. The depth of penetration. So blue light, yeah, that's a good point. So we did these things in it's like an LED light box, and then we put a plate on it. So it's whatever, the thickness of the glass, and we only monolayers. So our experiments, it's either microscopy or either these light box that we use, in the 96-well plate. So nothing fancy, but the blue light, about like tissue penetrations of red light. Red light, I don't know. I forgot. I mean, red light penetrates a little deeper than blue. Blue, it's as a few cell layer deep, right? So yeah, that's a good point. I don't really know. I never tried them in mouse yet or in animal yet. But they're not deep. Even the red light is not deep. So that's all I can say about this. So we want to see whether we can actually have orthogonal red light controlling one enzyme, blue light controlling a different enzyme, and then have it work, let's say in this, in the same cell in this circuit setting. So, so we actually have to find which pair works well together. So we use the AND gate as a way to, as a screening tool. So we ultimately, I'm not going to go into these data too much, but we sort of like having blue light control flip, red light control Cree, or vice versa. We have blue light control Cree and red light control flip. Okay. And we try all these different pairs as well to see, which combinations of them give us the best dynamic range. So we found a few, in general, blue light control Cree, red light control flip, for whatever reason, it works better. So we stick with that orientations. And then ultimately, we use our system to see we can control the output of the blade system right here. And then, so you can see here, so we can either have blue lights or red lights, and then we can have different outputs now from our, so this is the circuit architectures, and this is the outcome. So now we can use, blue and red lights to control the output. What's nice about using the blade system is that, ignore, don't look too hard at these little leakiness yet, but what it does is that, I have two inputs only, right? I have blue light and I have red light. Okay. I actually now created 3 unique outputs, okay, depending on the combination of the lights, right? So normally you can't really do that, right? If you have a simple reporter for Cree and simple reporter for flip, you have 2 lights, you have 2 outputs. Okay, even though you shine both at the same time, you just get 2 of them at the same time, right? Whereas in our system, if we shine blue and red light at the same time, we got a 3rd output, right? So given the fact that it's hard to have orthogonal light inducible system, so this gives us a new input, new output without having to reinvent a new light inducible system. So this is another test, of this like inducible reporter. So we're basically having, we asked, okay, we have two different genes, okay? We put them all in the same cell. And then what we did is that we can, let's say we do a photomask with only one side shine red light, on the other side only shine blue light, and then see how much of a, we also want to test like how sharp the boundary is a little bit. And then you can see here, it's actually pretty nice. I only see like mostly green lights here, and then they have a sharp drop off once, you're covered by the photomask, and then similarly for the blue lights as well. So it's pretty good. At least this system is quite clean, at least in this particular system, not the Myoblast. You can make it quite clean in terms of like pretty low leakiness. And then finally, we want to be cute a little bit. Can we also patterns? Have some patterns that's with the AND gate as well. So you can see here, we have to shine red and blue lights, and then it turns on. So it looks okay. And then you can quantify the systems as well. So so that's the story I want to share about our multi-chromatic inducible recombinase systems. So we use it to pattern cells, regulate cell fate, do logic circuits with it. So I'm going to switch gear, okay, completely from a mammalian cell to no cell, okay. Because I always thought like these systems, like nice thing about recombinase is we've shown that it works in bacteria, and obviously it works in mammalian cells. A lot of these recombinase come from phage as well. So it works. A lot of the biochemistry has been studied using them. It's in test tubes, so they don't care whether there's a cell or not. So it's quite versatile. So I always thought, yeah, it would be a great tool to use it in a cell-free system because many of I trust in this audience. There's a lot of use, case, interesting for fundamental study and for manufacturing in cell-free system. And I, the nice thing is that it's not alive, so you don't have to worry about cell viability and all these things, right? So there's no membrane to block things in and out, so you don't have to worry about transport of things and toxicity. You can use pretty harsh chemical. I mean, as long as it doesn't destroy the enzyme, the cell, there's nothing to die. It should have high sensitivity, robustness, and they're not alive, so they're not like not genetically modified organism. You don't worry about like releasing into the environment. So there's a lot of advantage of it. And I always thought that it would be great to have, these play system in a cell-free environment. So that it provides some tools for it. So it'll be great to have these tools, biocomputing in the cell-free systems. And so one of the things we try to do is, can we implement the Blade platform, especially these multi-input, multi-output genetic circuits in cell-free. I must say, I thought it would be pretty easy to do when I first started because of all these literature evidence, and then how far we have moved in mammalian cells, and we thought we knew this recombinase pretty well. So I was so wrong. I'll tell you why in a bit. So this is a work by Jingyao Chen. Sorry, I didn't have a picture of her, but she just graduated. She developed this system, on her own, pretty much. She called it the cell-free recombinase Boolean operating system, or CREBOS. So, so again, it's cell-free. So we do, we like to develop a system where they can orthogonals, they sense multiple circuits and set input and regulate multiple outputs. This multiplex sensing capability, and then we think it will be very versatile. So the first thing we did is just to see whether recombinase work in cell-free. format. And I thought, I thought someone has done it, but turns out it hasn't been done. But so I was very surprised, by that, first of all. And then, okay, that's fine. And then, no big deal. We'll do it ourselves. So we first let's see, recombinase works or not. So very simple thing. Let's say we use forgot to tell you, so we use these pure express system. I skipped the slide. So so those NEB pure express system. So we just have all the mixes for transcription and translations. And then we just put in DNA in there. So it has a T7 promoter, so initiate transcriptions. So we thought, okay, we'll put in a transcription termination because T7, these bacterial promoter, we just put in a bacterial transcription termination sequence. We flank them by the recombinase binding site, right? And then the reporter, we use mCherry. So if in the presence of recombinase, again, it will excise this transcription termination sequence. And now again, these are bacterial transcription termination sequence. But then it will allow transcription to occur. You see a reporter. And then we also, now this thing right here on the bottom is the troubleshooting. Okay. So we didn't anticipate we need to troubleshoot a lot. But anyway, so because we need to do some troubleshooting, so we also made this what we call a constitutive construct. So we were trying to figure out what's wrong with our system. So we have this promoter, and then just the recombination site, and then with the mCherry. Okay, so now without the recombinase, it doesn't do anything. It won't cut anymore because it only has one of the recombination sites, so it won't do anything. Okay, in theory. So here's what we think should happen. Okay, so look at this reporter construct. Without Cree, you don't have any output. With Cree, you have some output. Okay, we'll measure them in the plate reader fluorescent. And then now just this constitutive, we call it LOX-P mCherry construct. Okay, with and without Cree, it shouldn't matter. It's just, it should still have high level of mCherry. In reality, it does none of that. So first of all, look at this reporter construct right here. Without Cree, it has some basal leakiness. And with Cree, not only did it not increase output, it actually decreased the leakiness. We're like, what the hell's going on? And then, so when we see this, we're like, I don't know what's going on. So we start making this, constitutive LOX-P construct and see what's going on. So I mean, we have some hunch, okay, of what's. Again, we have without Cree, we have pretty high expression level because this is just like T7 promoter driving mCherry, so there's nothing special about it. But when you add Cree to the mix, it drops quite a bit, the production. So somehow the presence of Cree is actually blocking the transcription from happening. Okay, so this is baffling. Very humbling, actually. Like I thought it would just work, but even the most simple step doesn't work. So Jingyao, basically, what we've seen is like reporter has high basal expression, and somehow the Cree is negatively affecting the reporter expressions as well. So we basically have to completely redesign even the simple reporter system. So first thing we did was we try to figure out what's going on with the leakiness. So Jingyao found that a lot of people these like transcription termination sequence that people use aren't super strong. They're not really strong enough. So but there are known transcription termination sequence people have been optimizing. So this ultimately there's this double transcription termination sequence. So basically I think two of the termination 3 combined, then it gives you basically, it starts off with the original design right here, which you call it termination 1, and then 2, 3, and 4. So ultimately the fourth, basically double of 3 works the best. It reduces the basal by 94%. So okay, all right, good. one problem solved. Now we still have the problem that the presence of Cree not only does it not turn things on, it actually blocks expression. So it's like it does like exactly opposite of what we wanted to do. So we start actually we've seen this before. We start hypothesizing that the presence of the LOXP sites actually. So when recombinase, we need always a pair of recombination sites, and then it actually recombine, it forms back its own sites, but it loses everything in between. But this is still can bind Cree quite well. If I remember correctly, it's like an 100 nanomolar range. So so we actually think that the recombination site is sort of acting like a transcription a repressor sequence, and then the Cree becomes a repressor. So we thought maybe we can do something. There's a mutant lock site. So what it does is that you could engineer a pair of these like wild type LOXP and a mutant LOXP site. And then when it recombine, it forms this weird LOXP site that it can no longer, it binds to Cree in a lower affinity. So we tried that basically, and then we found that it actually sort of, so there's a 55% decrease when we use our original LOXP site, but then we created mutant LOXP site, then it doesn't bind to Cree as well, and then we sort of recover this loss in basal activity. I mean, basically we recover a lot of the expressions. Okay, so it doesn't inhibit as much, but still, okay, we got rid of the sort of the inhibitions by Cree. We still haven't solved or one of the most important goal is to actually, when we have Cree, it'll actually increase expressions, right? So what we found is that we actually made a mistake. Not made a mistake. We didn't know, like, how close should you put the recombination site next to the promoter. So if you actually place them too close to each other, they sort of actually blocking each other, because the T7, we think the T7 and the Cree sort of like interfering with each other. And they are not we actually think it's either the T7 is preventing the recombinase from doing its job, or vice versa. But if you actually space them away from each other, then we can now recover the inducibility. So before, I think we were doing it like right at the end of the transcription, the promoter, we put the LOXP site, and that was not good. So you just space them out like 10, 20 base pair away, then you can recover the expression. But if you space them too far away, then it won't, the transcription won't work. So we went further than 30 base pair. So ultimately, so even designing the promoter took us like a year to figure this out. We went from like actually having high basal, and then the Cree actually reduce expression, if anything, to so basically from like 0.2 fold, it's not even an induction, it's inhibition, to a 41-fold expression. So the changes is quite a lot, right? More than 100-fold. So now we finally built a reporter for recombinase in cell-free system. Then we're using the same kind of design principle, so then we use that for other recombinases as well. So we have Cree, this 5C, and these other recombinases. So that's nice. And then once that start working, we're like, okay, we can probably use try the Blaze system in the cell-free as well. So it looks okay. It has a little higher basal on the last lots, but it looks okay. Not great, but not bad. So now we also have Blaze system working in the cell-free format as well. the next thing we try is, again, we don't, you don't want it to be just, you want it to sense something, right? The whole system can sense something. So we ultimately end up using the system from Julius Luck at oh yeah, Regina, sorry, I just got your message. Yeah, we use peer system, not lysate. So we use peer. So so this is the system that Julius Lux used. We borrowed his proteins, and so the way it works, you have the transcription, you have the promoter, and then he has these basically transcription factor binding sites that normally it'll sit right there and block transcription, and then if there's a small molecule, it will de-repress and allow transcription to occur. If you're curious, we try really hard to use these split dimerization system in cell-free. It does not work. I don't know why. There's a lot of things I don't understand in cell-free. So again, it's quite humbling. Yeah. But so we end up using two systems. We screen a few. Not everyone works well, but we end up using a tetracycline inducible one and a zinc inducible. Try either it's control Cree or 5C, and then we can show that we actually can regulate blade circuits doing that. So we like to use the blade system because it's one of the more complicated circuits. If it works with blade, it works with other types of circuits as well. So one nice things about recombinase I always like, and that's why it's used in animal studies a lot, it's because it permanently modifies the DNA. So it has this basically natural memory capability. And we know that a lot of these complex systems, being able to record event is really important, So we're wondering if we can actually use the cell-free recombinase system as a storage a device, right? So DNA has become, it's a natural biological memories systems. Of course, there's a lot of people are thinking about these things, right? And because it has high stability, yeah. Mohammed, yeah, I don't know with the self that I don't know. I have not seen it with recombinase. We try with recombinase, it doesn't work. Maybe TFs, I don't know. I have no idea. So yeah, high stability, high storage capability, density, you don't need, power consumption is low, right? It can you can copy, you can inherit to the next state, and easy to read and write. It depends, you can always sequence them, but there are like restriction enzyme and ways like you can tell whether things get modified or not. And then it's environmentally friendly, it's easy, it doesn't take a semiconductor plant to make a memory a DNA device. So so another thing that we're really interested in is that you can store DNA, on paper and lyophilize it, pioneered by Jim Collins, right? So we thought that would be great. So you can freeze dry, a lot of these, circuit components right onto paper, and then right, perform your reactions, sense things, and then right, it changes it, and then it stores in the paper and DNA. And then when you need it, what we end up doing is actually we loot the DNA out of the paper and then transform them back into bacteria, and then see if it gets modified. We can sequence it, and right. So that's what we did. So at first we tested out. We want to see if we have these reporter with Cree, and then we have in this paper-based format, how stable are we? Can we recover the DNA and then basically to recover that memory that the Cree reaction has been occurred. And so it'll last for like 120 days, right? So it's not surprising. We know that DNA can last for like years and years and years. So and then finally, this is the last figure in the paper that hopefully will come out in 1 to 2. So we also shown that, let's say you have your circuit, it senses things, either TET or sync. And then in this paper-based format, right, can you remember the DNA, the input, right, the logics of these things? And then, of course, it modifies the DNA. And then we actually, can we, recover the output, basically. We run the pure expressions again, pure express system, and then see, can we generate the output. So at least in this particular system, right, you can remember, at least for 28 days. So we envision that there could be a lot of use, initially it's designed for like the self environmental monitoring. We have a little device, maybe in some microfluidic devices, and then we can sense like environmental factors. It triggers the the expressions of Cree or FLIB or 5C, right? And it modifies the DNA. Okay. And then we don't need to read anything yet. But then someone, later on, people can come and retrieve the device and then see if the DNA gets modified, right? They can either, sequence the plasmid or run, elute the DNA out and then put it in pure express and then measure the output, and you will know, what has been happened before, right? What the system has sensed. So that's what we imagine these systems would be. It's a low power system, and it remembers, it records the events. So with that, that comes to the end of my talk. I just wanted the first part on the tissue patterning, the light inducible multichromatic is done by Christina et al. She's recently graduated. And then the cell-free system is done by Jing Yao. It's in close collaboration. So with Doug Densmore, actually, for the cell-free system, and Christina's work, it's in collaboration with Chris Chen and Daryl Cottons, and these are the funding sources. So that's the end of my talk. Free to have questions.
Thank you so much. I really like it. Obviously, I really like the Cree work the best, but everything was really cool. There is several questions in chat. Can you still see chat?
Yep. Okay, let me see. Fantastic. Did you ever try to do cell-free work in liposome? No, I have not. Yeah. What is it? Yeah. What can I do with it? So can you share a bit more, like how difficult it is to replicate forward channels, genetic circuits using these recombinase and cell-free systems? So the play was okay. Once we figure out how to even get anything to express reasonably well in a cell-free system. So I would say initially the first part where we optimized the reporter system, that took a while. Once that working, it was okay. It wasn't too hard. To make it inducible took us a long time too, because we were trying to do the split dimerization system. We tried light, we tried chemical. I don't know why it just doesn't work. So yeah, I was pretty bummed by it because it seems like it will be very versatile, very useful, but I don't know why it doesn't work. So have you tried using mammalian cell-free extract? No, We have not. We never tried to use. We tried to use extract in the beginning. It wasn't. I don't know. Either we don't have consistent batch of extract, or yeah, it just didn't get very good result very early on. And then my Jing Yao wants to graduate. She doesn't want to spend too much time, so she's like, okay, it's just too expensive. Oh, okay, that looks expensive, but so that's all we did. I'm open to trying with lysate and extract again, but we just didn't have a good batch very beginning. Okay, Hannah asked, did we try more channels? We are trying a green one. It's not working. Martin Fusinager published a green one. And hasn't been working for us yet with recombinase, so we're still troubleshooting. And there's not much more wavelength. There might be a UV system. We're reluctant to try too much because it fries the cell. So, there's not a whole lot to choose from. Maybe a green one. I don't know if there's an infrared one. I think there is an infrared one, but there's too much overlap in terms of their excitation wavelength between a lot of systems. So up to a certain point, you're gonna start seeing a lot of leakage. Yeah, we switched to nanoluke. Yeah, mostly because some of these IRFP requires like cofactors and things like that. I don't even know if our plate reader can read like far red well. So I think it's just a technical limitations. The optimized sequence for the 10 is published yet for the 10 base pair gap. Yeah, that's all the sequences will be published. It's in Cell Systems right now. We have all the sequences in the paper. So yeah, it will be published.
I think that's all the questions. I just want to say thank you because I really appreciate, especially the cell-free system work. The working with pure system is really challenging. I really appreciate all.
I have I thought I know how to work with recombinase pretty well. And I thought, oh, it's a cell-free system. It's like bacteria. It works in bacteria. Other people have shown it, but it's not. So yeah, it was yeah, it was much harder than I thought. So I'm glad it worked out okay at the end. But at the beginning, I was like, oh goodness, why it doesn't work?
Yes. Yeah, that's a common sentiment. I hear people getting started on especially project in Pure, and it seems like it should be working because all the parts are there, and then you bang your head against the wall, and then 6 months later you finally figure that out.
Is it better in lysate then?
Many things are better in lysate, at least in our experience. One of the things that's really working against you in Pure is that the reaction yields are relatively low, so you don't have much room for error. We got stuff to work in lysate that we couldn't get to work in Pure.
Really? I would love to try in lysate. I mean, we got some lysate from Doug Densmore. And then, this batch variations, and then yeah, my student just gave up really quickly early on. They didn't want to deal with the lysate. I would
That's an
love to get
unfortunate. Yeah.
I would love to get a consistent batch of lysate and try it out.
Not just you. We all would love a consistent batch of lysate. The batch-to-batch variability is a huge problem.
Really, really.
That's
Yes,
not solved yet.
I would love to try the split dimerization recombinase that we have, because there's a huge number of inducible elements we could try using that. But without it, like I can sense glucose, you know, if I can do split dimerization, I can sense red light, blue lights, right? There's more I can do, but I mean the transcription factor that Julius Luck has, it's also pretty good. It sends, you know, some sync and, you know, so those are pretty good tunes. But having more would be great, would be powerful, I would think, in the cell free.
Yeah.
Yeah. Everything I do will be available. Shoot, I should put. I'm planning to put everything on Agile. Now the student left. Now I have to do it.
You better deposit it because you're gonna keep getting emails, people asking you for plasmids. It's
Yeah, I do. It's my goal. Everything we publish, you know, especially in these genetic circuits, to be on Agile.
Yeah, I have the same problem. If I get lazy and don't deposit, then people start emailing, and I'm like, oh man, I should have deposited that.
Yeah, I always make my student do it because they will know where things are. They will know which ones to put. I don't, you know, I don't know where your freezers.
Yeah.
I get lost in my own lab. I don't know where things are. Okay.
All right. Thank you so much. Thanks for a fantastic talk, and thanks for a great discussion.
Yeah, thank you for inviting me. This is fun for me. Thank you.
And thanks everyone, and have a great rest of your week.
Yeah, thank you.
Bye.